what we've seen is that when we expose mammalian and especially human cells to our very precisely designed acoustic sequences, that's how we call it, we can make their membrane more permeable.
Hey, smart biotech scientists, welcome back. We are in the middle of a conversation with Gabriel Dumy, who's the co-founder and CTO of Kolibri, a biotech company aiming at redefining the mammalian cell's bioreactor through the use of acoustic waves to sort, to engineer and grow these high added value cells in the best manner. And if you haven't listened to part 1 yet, please go back, because today we're continuing our exploration of the use of acoustic waves to revolutionize cell manufacturing. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Now let's look at the harvesting. You mentioned that you can use your technology to harvest as well. How does this work?
So what's interesting, you can think of the acoustic wave structure that we built as a scaffold, but that would be reconfigurable in real time. We can control it in real time, move it up, down, increase its trapping force, or release it. And so, for instance, if you are working with adherent cells, they are not stuck into a very remote location anymore. You don't need to trypsinate them, damaging them in the process. You just have to cut the acoustic waves, and you can literally harvest them directly. And so, for everything adherent cells related, it's very useful and very improving the situation of the harvest. Another aspect of it is that because the cells are trapped in very specific locations, either you can harvest the supernatant very easily because you will not have cells, debris or contaminant going in. And if you want to harvest the cells themselves, such as is the case for AAV, for instance, then you can trap them in a location where you will harvest only the cells, making it easier for all the DSP processes because your sample will be pre-concentrated if you want. And last but not least, we are studying it these days. The capacity of the waves to actually crush the cells, to break them open, can be used in certain lysis steps. Instead of using a detergent, you could actually apply a very specific acoustic wave to your cells, and it will break their membrane open, thereby allowing you to harvest anything that will be inside, whether it's AAV or another biologic that you would be producing. So it's a very interesting alternative to your usual stirred tank because you can actually do more with your acoustic waves than only stirring. You can actually perform convoluted steps that are hopefully helpful to the bioproduction you're targeting.
Speaking of doing more, Gabriel, let's move very upstream and let's look at the transfection, because this is also something you can do with your technology, right?
Yes. So that's something, as I pointed out, that is a block in a lot of gene therapy manufacturing today. But even if you're looking at cell line generation, when you want to modify a cell, whether it's to modify it in a transient manner or a permanent manner, you still need to deliver a cargo to the inside of your cell or to its nucleus. And notoriously, it's a hard thing to do. Depends on the cells, of course, but it's always complex. And what we've seen is that when we expose mammalian and especially human cells to our very precisely designed acoustic sequences, that's how we call it, we can make their membrane more permeable. So basically, we can deliver a cargo much more efficiently from the supernatant to the inside of the cells. Today, we have tested this out with AAV cargo, basically, so to 4 plasmids with transfection regions, but we've seen significant improvements, basically using between 2 and 10 times less plasmid to have the same cell productivity in the end. And I'm sure what we have here is in fact a kind of sonoporation device, basically, that we have optimized for gene therapy today, but that could be used for any cell engineering that you would do. If you want to modify a stem cell, if you want to modify a human immune cell, for instance, the fact that we can, in a very controlled fashion, make their membrane more permeable will actually be useful to a lot of people, not only to gene therapy manufacturer. So that's still something that we are optimizing today, but we've seen that we can actually help the transfection processes in gene therapy manufacturing.
And what about the transfection efficacy? Is it similar to traditional methods or is it very different at the moment?
We are on par. What we are trying to do, because it's a very new tech, is to compare with the most standard system we can find. So we have very standard AAV2 and AAV6 vectors that we are producing in very standard HEC293 that we buy from Thermo, that we buy from ATCC and so on. And so what we see when we compare with, again, standard protocols is that we are either on par, so we are almost never performing less, but often we are above the limit, basically, the control, in terms of vectors production and in terms of vectors infectiosity. So basically, the vectors that are produced with our methods are consuming, so that's what we measure, less plasmid per cell. We interpret this because we have a much different cell approach, shall we say, and a cell exposure to the plasmids themselves. And the vectors that are produced afterwards are competent, basically. That was one of the key points, because if you improve the transfection by 10x factors, but you produce only empty AAVs or non-competent AAVs, then it's basically pointless.
Now let's shift gear a bit in this final part of our conversation, because you're developing a new technology and this requires a lot of prototyping. So can you tell us a bit your strategy, how you do that, the approach you're following to make this efficient and to get the best possible outcome?
It's actually very interesting to talk about these topics because it's usually overlooked by either startups, CDMOs and so on. We started from scratch, like zero, paper ideas. And so we started from scratch and we decided to go on viral vector manufacturing, which is actually quite complex. And so we are still here today, 3 years after. Good for us. But it was a journey very rich in learnings. And so what we did was the following: build as quickly as we could proof of concepts bioreactors, very small ones, but where we could actually de-risk, so show the world that the cells would be okay, that the cells would be producing, that the production would be valid for an industrial use. And then with this data set, find the financial, the human resources, the labs, to basically upscale it a notch and do it again. And if something in the process tells us that either it's a bad idea, it doesn't work maybe, or the market is not ready, or it doesn't perform as well as we thought, well, basically pivot, check another biologic, check another cells, and see how it goes. But the goal of this company is to bring a solution to the bioproduction issues we talked about earlier. Maybe this solution will be in the shape of a transfection module for gene therapy. Maybe it will be in the shape of an adherent bioreactor that performs better. I don't know today the answer, but that's the goal. And so in each iteration, we have our prototypes that we build internally. We try to generate an interesting acoustic field, and we test it with our bioproduction team. So today we are testing a lot on AAV production. So it entices to have in your hands as many of the QC that are related to the biologic you want to investigate. So we have PCR, we have assays to understand as quickly as we can what happens to the AAV we are producing. And basically, if we need to pivot, the strategy so far is to do it through partnerships. Today, we have a tech that's stable enough that we can imagine bringing the machines, 2 or 3 of them, to an academic lab that's doing, iPSC, or to a CDMO that's doing Lenti, for instance, and to test out like these other biologics with this partner and not needed to internalize like the very precise skills that you would need to produce a Lenti vector, to characterize it, and so on. And so the goal is to increase the scale and the maturity of the technology that we have today. Basically to move from a plastic lab-grade bioreactor to something that's cleaner, that's GMP grade, and that's bigger over time. The final scale will depend on what is the commercial success, whether it's like development AAV bioreactor or super large-scale cell harvesting module, for instance. But that's the 2 pillars of the roadmap, basically increasing the quality of the machine and increasing the scale on which it's working. And of course, the 2 pillars involve a lot of R&D, IP, communication with this very conservative industry, because that's the one thing that we'll need to do if we want to have adoption at some point, is to produce again and again with partners that can actually vouch for the tech, that can say, look, it's not as crazy as it sounds. It's actually working on my cells. It didn't introduce a contaminant. The biologics are performing well in the end. And so it's basically proof casing the tech that will hopefully make it a success one day. So to sum it up, it's a lot of internal R&D, spinning out partnerships to go faster on the de-risking, and then communicating about this de-risking.
Yeah, smart biotech scientists, this is excellent advice. Get early feedback or as fast as possible to be able to pivot. And now a question that comes up, I'm advising different startups in my role, is how early do you go in front of partners? Like how minimal should a solution be, or can it be? Or how long should you wait?
I would say it depends a lot on the partner. For instance, you will not address an academic team as you would a large pharma like Merck or Thermo Fisher, for instance. Both will have very different mindsets. Both will have very different means. And maybe you will not ask the same thing. So it will depend on the partner. But to have a minima, an idea of what you want to do is paramount. Like, do you want to produce a white paper? Do you want the partner to buy one of your machines? Do you want to have a long-term collaboration because you will produce for your partner biologics through preclinical phase 1 to 3? I don't know. And so if you want to de-risk, for instance, an application and you go and see an academic team, usually these profiles are very open, very eager to test things out, because that's how they can actually generate knowledge and publish about it. And so often you can go see them with a proof of concept, or even an idea of what the proof of concept would be on their cells, on their biologics. So that's what we are doing. We go and see academic groups with the idea, hey, we could produce your very hard to grow stem cell with our machines. And basically at the end, ideally, you get a way to grow your cells in an interesting fashion. You get a paper, and we get coverage, a white paper, and so on. Conversely, if you go and see an industrial, whether it's a CDMO or a pharma, these people are on an agenda. Their time is often precious, and they usually have much larger capacities than you. So you need to have your IP right. Basically, if you are bringing them a miracle machine that works very well, but that they can copy at will, they will do the partnership very quickly, like often suspiciously quickly, but you will lose your IP at the end. So have your IP ready when you want to do industrial partnerships. That's step 1, I would say. And then, because time is precious for them, but also for you, define in advance the framework of the partnership. What do you want to get out of it? What could they get out of it? And have the key de-risking point tested out in-house. Basically, if I want to produce a Lenti vector, I will try to do very basic assays in my lab with the right cells maybe, or with the different plasmid kit, just to show that it's not like a huge risk for them that we can actually try the tech on their cells and they will not totally fail, and hence not a totally waste of time for both parties. And when you go, like the larger you go, the more stringent the asks will be, like more data, a better machine or a larger machine, and so on. And it's very hard at the beginning, I will not lie, because you have very little. But over time, as you train, as you get more used to it, and as your machine and your data gets better, it's usually flowing with more ease. And so it's very much like a training, like sports, like exercising. Like you get better at it, the more you do it. The feedbacks you collect are improving what you do. And so the first steps are the hardest. Like the first partnerships are the hardest. Usually there are failures. It's okay. It's part of the journey, as long as you improve and you learn on the way.
Now, looking ahead, Gabriel, what breakthroughs do you envision for acoustophoresis in biomanufacturing?
So as I said, it's a tech that I'm in love with ever since my studies. I'm really convinced that it has a lot of applications in biomanufacturing because it's a low power approach. It's very compatible to human cells and it's scalable, basically. So far, we've only seen, let's call them micro to millifluidic applications in cell sorting. For instance, you have the Attune module cytometry, which is aligning cells under the laser. We at Kolibri are trying to upscale the tech to do cell culture, cell engineering, and so on. So I think the breakthrough will come through the fact that you can use it in a very effective fashion to pattern, to enhance, to simulate the growth of fragile cells. So probably at the beginning on small scales, but probably on much larger ones. And that's still academic today, but we see more and more research groups that are growing organoids, for instance, with very small acoustic fields, or patterning cells. So I see huge breakthrough in complex tissue engineering, for instance, or in co-culturing how to grow cells when you want to grow a cell that needs a protein or that needs a feeder cell line, for instance. And basically, in every bioproduction step where you need a precise and non-contaminating control over your cells. When you want to do cell washing from medium A to medium B, when you want to do cell concentration because you want only the supernatant or only the cells, or when you want to do like cell modification where you cannot afford a loss in viability. These are all the very hot issues today where acoustic manipulation could be an answer. Whether it will be the answer that the market adopts or not, well, it's not that easy to predict, but that's where the tech fits the better, with very high added value fragile cells that you want to edit or to grow in a controlled fashion.
Gabriel, what is the most important takeaway from our conversation today?
So I would say that the most important thing to recall from everything we talked about is the fact that bioproduction is a boiling field, that it's treating and addressing very important points. Pharmaceutical bioproduction, which we are pursuing, is especially important to me, and that it lacks today a lot of crucial tools to be efficient. And these tools are found in the interface between biology and other domains, whether it's chemicals, physics, engineering. And that if you are looking for a challenge and that you are fluent in either biology, engineering, or both, that's a place where you could have actually a huge impact on the world. It's a domain that is necessarily very conservative because it's pharmaceutical in general, and you need certification, qualification to push an innovation forward. Hence, the fact that people have used and reused a lot of tech that was working fine, but that is outdated in order for these very promising technologies to be produced for the greater people.
Where can people connect with you?
There are several channels where people can actually reach me out. The number one would be through our company's website. So it's www.kolibrilab.com, KOLIBRILAB.com. There is a contact form that you can use. You can reach out to me directly. It would be Gabriel, Gabriel, @ kolibrilab.com, same thing. Or you can even reach me out on my social networks, so LinkedIn and ResearchGate, where I'm usually particularly active. And that's about it, I would say. And if you are in Paris, well, feel free to swing by. We are in the city center on brand new labs, and I would be pleased to show you the tech and give you a tour.
Yeah, smart biotech scientists, please make sure to reach out to Gabriel. I will leave all the links down in the show notes. And Gabriel, thank you so much for sharing these insights and being on the show today.
Well, thank you, David. It was a pleasure. Thanks to you for hosting me in this show. I'll be sure to listen to every following episodes.
Thank you for tuning in today, my fellow scientists. Let me know what you think about this episode. And perhaps you have a breakthrough technology yourself and you're wondering how to bring it to the people who most need it. So let me give you a very practical offer. Use the show note below to book a free 30-minute consultation because I want you to help get started. So book your call now and I see you in the next episode. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.